My name is Edward Park, and I'm very grateful for everyone showing up on a Sunday. I know it's very hot. We have the engineer working on that. I hope I can share something that you find interesting today. So how many of you actually got the email from me, the welcome email? That's so hands. Okay, so about a good half. I kind of explained how we got here. It's all very new, very exciting. So I want to explain to you why I think exosomes are the future. And you guys are at the very cutting edge. This is an electron micrograph of exosomes. These little spheres are smaller than viruses, so they go everywhere. And this is actually, we found out over the last 10 years, this is how all cells communicate. So when a person has cancer, the exosomes are bad. It's like if you take a good person and put them in a bad situation, they become bad. If you put a bad person in a good situation, they become good. This is how the cells are talking. This is the music they're playing to each other. Okay? So, the first 2 minutes, I'm going to talk about the story of how we're all sitting here, and then I'll explain cycloestranol, which is what you got hopefully in your bottle. Who didn't get a bottle of Recharge? Oh, good. Okay, just one. So the break will be easy. Everyone's got their bottle, and the instructions explain how to take it and what to expect. Okay? And the lecture will go about 15 minutes. We're going to have a break, and then they'll bring in some food, some fruits, and cookies. And we can chit-chat a little bit. Ever did your parking validation? Oh, this is great. Oliver did it. My son Oliver did a great job. And Chika-san as well. So, and then we might get out of here early then. We're going to do a clinical lecture at 3:30, which is more like, kind of like, I hate to say sales pitch, but it's like if you wanted to actually get into exosomes and take them, what I've learned so far. And I've only been doing this for, 9 months. So my mom here, Youngju Park, go ahead and stand up, Mom. She's 82. She's gotten 8 injections. She was the first person to get injected. She's got 8 injections. So she did her face, and I allowed her to do my face with microneedling. We did her lumbar epidural, IV 3 times, nasal 3 times, right? So, and hands. We did a nerve block. We did her arthritis. Shoulder too. She's basically my pincushion. But I also do my own cooking. I've actually done my knee, my shoulder, my nose, and so far so good. So we'll talk about that more in a second. Okay, this should give my mom flashbacks. Okay, Mom. What's your favorite? We all know. Anyway, so I'm in the middle, I'm the baby. That's my sister and that's my brother. Yeah, so I was born in Canada in '67. When we were 3, they moved back to the United States. There wasn't any Asian immigration until Nixon, so my dad and my mom met in Pennsylvania, and they had to move to Canada when his residency was over. So I was born in the tundra of Saskatchewan. After 18 years of tiger parenting from this lady, she pushed 3 kids into Harvard. She's been writing on that for the rest of her life. So then from there, I went straight to Columbia to get an MD and a Master's of Public Health, and then back to Harvard Teaching Hospital for OBGYN. I practiced OBGYN for 4 years with Kaiser, great organization, and then 10 years on my own. In 2007, I took something called a telomerase activator, and it really changed my body. It took me out of metabolic syndrome. I had high blood pressure, fatty liver, high cholesterol. And John, I know, was just about at the same time, 11 years. John, will you stand up, please? This man, Chuck Norris body double, he does all the stunts. So John, he came from New Zealand. He's been taking T65 for 11 years. I've been taking it for 12 years. And as a result, all original black hair, no reading glasses, I can read this close. I used to have gray hair 10 years ago. So, I have a lot of patients, a few dozen, and for whatever reason, it seems to be working, I don't take any medicines. I used to take medicines. I do not. What you have is 25% more, and I make it myself, and it's very powerful. So the instructions are something you should read. So, because it affects your sleep. You can have waking up between sleep cycles, and that's all explained. So ever since I started taking it, my patients would come back with their pap smears, and they'd say, well, why do you look so different? And I said, well, I don't know. And after a year, I said, okay, I'll start to prescribe it. And when people would tell me, oh, Dr. Park, did your diabetes ever get better, or your libido, or your, whatever, cancer? I would say, no. But when 2 or 3 people said the same thing, I would do a podcast. So there are 50 podcasts, over 500,000 views on YouTube, teaching people about the disease, and then interviewing live people who say they got a little better on T65. I mean, I'm very skeptical by nature, but you have to be open-minded too. So, the testimonials are interesting, and they're there to let you know. From those experiences, I wrote a book, self-published, and then last year I was asked by Hay House, they do a lot of self-help books, to write a book on personal health. So it just talks about the 6 areas, breathing, mindset, sleep, diet, exercise, supplements, what the science is. Very skeptical, but I think there's a lot of stuff that you can learn from there, and you can order that on Amazon. So the reason we're here is because of this man. Almost a year ago, December 10th, I saw this guy. He was in one of those, those half-kneeling skateboard things when people break themselves. And the owner of the big Las Vegas conference, Anti-Aging, was also on crutches. He was pretty sick. So I was laughing. I'm like, these guys are anti-aging doctors. They look really sick. But when he got up and told his story, apparently this is a 64-year-old, at the time, neurologist. And he laid down his bike, broke his clavicle, 11 ribs, his ankle. It's just crazy. I said, so I'm sitting there thinking, like, is this guy a liar? Like, I had a tear in my muscle. It hurt to even laugh. And he's up there telling stories, feeling great. He lost a third of his blood. I said, okay. In my mind, I'm thinking, this guy's either a liar or there's something to this. And so the story was that, without telling his doctor, he would already experiment with exosomes. He took 5 vials and injected himself IV. You didn't miss much. So anyway, his blood count, he had lost a third of his blood count came back. This is his ankle. I'm thinking, like, I'm listening to this story, I'm like, is this guy, thank you, Mary, I'm gonna wear this. This is his, the doctor's ankle. It's fractured there, and then it's pinned. But it's not a good resolution photo, but don't worry, you can see that a month later, and this is a day or 2 before the lecture, there's really no hairline fracture there. So something incredible and healing might have gone on there. And so I thought, And then I listened to the founders of the company and the stories they told. Granted, this has only been out for a year at that time. Very early. I said, wow, this is something. Now, these are just pictures I sent you. That's me in 2000, when I was 33. I was about 10, 15 pounds heavier. I'm gaining the weight back now, but very sick. Just taking the T65 seemed to take me out of that within 3 months. No diet or exercise. After that, I told you I prescribed, and I started to learn what happened. So what you have is 10 milligrams of cycloestradiol. It's also got 3 herbs, gynostemma, holy basil, and ashwagandha, which are adaptogens. They might be telomerase activators, but basically they probably trigger the endocannabinoid system, which is the system that triggers when you feel happy. You get better sleep, vivid dreams, faster recovery from exercise, hair tends to grow faster, sometimes darker, skin and nails, fewer colds and flus. These are just common things. Some people say they need less caffeine and their mood gets better. It's funny, I didn't plan on being in Hawaii. My son, who checked you in, wanted to go to Iolani, so that's where he's at. So the reason we're at this lecture is because he's here. So, Lokahi is actually a Hawaiian word. I looked in the words like Hindi and Native American Chinese. I wanted to find a word that meant balance, because actually good health is not just absence of stress, right? If you lie in a bed, your muscles atrophy. If you have no challenges, no worries, then your mind gets soft. So good health is to be able to react and maintain equilibrium despite internal and external stresses. The number one source of toxicity is actually from our own bodies. If we hold our breath for 2 minutes, we die. If we don't eliminate through urine, we die. Toxicity is a constant part of life. So good health is when you are in equilibrium, you're balanced. Okay, so like this story. So my good friend, who I didn't know a year ago, his name is Duncan Ross. And I want to tell this story because I think it really shows how we got here. So Duncan Ross, he tells a story about his dad being sick, and he wanted to cure his dad. So he got a PhD in immunology. He was accepted to med school. And around 2012, he founded a company and he started growing people's stem cells. Now, a lot of you might know people who did stem cells. And we can talk at the end about this at great length. But the point is, they take your fat, usually, and they try and expand it. So you take one cell, you divide the whole colony, you get millions. So he was trying to cure this plastic surgeon's wife. She was 70, but her cells wouldn't divide. Why? Many reasons. So he couldn't do the autologous stem cell transplant. So he took the serum from an 18-year-old paralysis victim, he put it in the old lady's cells, and he started dividing. He said, oh my God, the old cells can react to the young signals. And that's when he decided to shift his company from a stem cell company to an exosome company. And that's what we're getting into. Same year, a couple months later, he was at a Halloween party, and this friend of his threw water on a gasoline fire. And so he got these terrible burns. I want you to pay very close attention to these pictures. They don't reproduce well, but you see the white patches, that's where the burn is. The darker skin is okay. Okay? So he took this serum with the exosomes we now know, and he said, buddy, don't let your doctor put PRP. It's going to make it worse. It's going to cause scarring and inflammation. And African American people get keloids and it can be very bad. So he convinced the plastic surgeon to let him do it. This is day one. You can see the red, the spots. Remember the spot there that's pale. Day 7, he's just topically applying the exosomes, whatever the stem cells were excreting. Billions of those. 6 months, he looks pretty good, right?
Yeah.
Great. Now the point is, this picture. It took me a while to figure out the picture. He never really explained it. It looks Photoshopped, doesn't it? It's not. It's not Photoshopped. What it is this is the normal skin that wasn't burnt. That's the skin that was burnt and given exosomes. And his point is that there's 38 collagens. That looks like baby skin. That's the same face. So the part that was scalded and burned and given exosomes comes back like a baby's bottom. I think that's kind of remarkable. And again, proof that old cells can respond to young signals. So this is going to be a little sciencey, and I want everyone took high school biology. I want you to think back to what we learned. So there was endoplasmic reticulum, meaning the mRNA gets shuttled into here, and they talk about this thing called the Golgi apparatus, and then they kind of just didn't explain what that was for. So scientists are now understanding that what that was for is that instead of just squirting proteins into the blood and hoping all goes well, they actually package them in these little exosomes. This is the main way we misunderstood all of biology. So what's going on here is a multitude of things. First of all, your lysosomes are destroying the internal tract. But the bottom line is, if it wants to drink in what's outside, it does these hits and it sucks it in. But most of what the cell is doing to communicate is not just, throwing out letters as paper airplanes to the world. It's actually packaging them in a little discrete package with hundreds of non-random specific messages. This is how they're talking to each other. Okay, so if you look at the literature, which I'll show you a couple articles, most of it's basic science. Over the course of the last 10 years, especially 4 years ago, it's been a hockey stick. Thousands of articles. But the bottom line is, this is how cells always talk. We just ignore them. There are 3 types of cells. There's the big ones, which come from a disintegrating cell. It's called an apoptotic body. Microvesicles, 100 to nanometers to 1,000 nanometers. What we're interested in is this. These things are called exosomes. And they're very specific. You saw them in the opening slide. They're spheres filled with specific 3 or 4 things. I mean, there can be some lipids and DNA, but mainly it's mRNA, messenger RNA, proteins, and blocking messenger RNA, which we'll go into just a little bit. And we know that they're exosomes because they have these special proteins, 9, 63, and 81. It's easy to remember because 9 times 9 is 81, and 9 times 7 is 63. And that means they're special, they're not random, and they're made to do something. Here's an actual picture of what it looks like. They come in these little, almost like a pomegranate seeds, or like, there's not many of them. They're little bags of little tiny spheres. And you can see it being extruded. So this is how it kind of just burps out these exosomes, and they go out and they are protected. Because the whole key is if you just send out proteins or RNA into the blood, it'll get destroyed. It doesn't last. So this is how the cells, and so what happens is these tiny spheres, smaller than viruses, when they touch a cell, they might bind by the protein, but basically when like the membrane touches the cell membrane, it just dissolves in, and that's how they talk. Okay? So it should have been obvious, but we always thought, oh, the proteins are just being excluded. No, they're not, because there's proteases, there's RNases. They have to be protected to get to where they're going. So why do they exist? This is how the cells talk to each other. The song depends on who's singing and what's needed. So what we've known for a long time is that stem cells, especially mesenchymal stem cells, are they go everywhere, they do everything, and they can differentiate, which is shown in this next slide. If you want to learn more about exosomes, I did 9 videos while my sons and I were in Hawaii. When they were hiking the canyon, I was narrating these videos. There's 9 of them, and it kind of explains in that basic science way what we're talking about. Is everybody with me so far? I haven't lost anybody? So again, because of their size, they go everywhere. There's no blood-brain issue. They're in blood, lymph, CSF. They have 3 things, and I like analogies. I liken it to gas pedals. We got the accelerator, which is proteins. The proteins get into the cell. They do what they're going to do right away. They have a brake, which is called a microRNA. A microRNA is a new class of RNA. It's an RNA, but it's a specific blocker of a sequence. So if you have a protein that you don't want expressed, the microRNA will bind the messenger RNA. Let's say you have some bad messenger RNA in your cell and you want to block that. The microRNA is a specific matching sequence that gums it up and can't be read. Okay? Okay. And then the messenger RNA is the clutch. So the messenger RNA is it's not just a single use like a protein and it hangs around until it's dissolved. The messenger RNA, you can see it, it's a message, it can be read by dozens of ribosomes at the same time. So a clutch, changes gears, makes it that you go 120 miles an hour. So this messenger RNA will stay around for weeks and transcribe thousands of proteins. Okay? I see a lot of puzzle looks, but you don't need to, it's just an analogy. Basically, it's good to get a protein. Like people inject protein, they go, oh, it's great, I got a protein, GDF11 or whatever. It only lasts a few hours. If you give the message, it's like teaching a man to fish, right? It's like that cell can use that message over and over again. But very important, it's not a cell itself, and it can't cause cancer. It's not a retrovirus. It's not going to introduce new genetic information or programming. It's a thing that lasts for weeks. Okay? So even across species, these things are respected. You see the mouse studies I'll show later, they use human exosomes to treat mice. I mean, someone was telling me last week, well, I'm worried about cancer, doc. I said, well, it's not really what you think. Cancer is not what you think. We have the same genes of 99.6% with chimpanzees, and all of us in the room start out with almost all the same genes. So cancer is something that just happens wear and tear. Like if you have a car for 500,000 miles, it's going to get bad. That's just the bottom line. There are exceptions. People who come from genetically isolated populations have single gene mutations of bad cancer genes like the BRCA, but that's the exception to the rule. Most all of us have good genes. Here's a picture of what MSC is, a mesenchymal or mesenchymal stem cell. They're these little cells, and they sit on the capillaries in every organ of your body, and when something is needed, they listen and they start sending out signals. And that's what they do. If need be, this is from my first book, Telomere Time Bombs. At that time, my thinking was like, well, this is actually science. They can differentiate into all different cells, bone, nerves, cartilage. So they're actually very poorly named. But it appears that the effect is not just from engraftment or differentiation. They actually go and they generate repair. Arnold Kaplan is the head of this whole field, really. He kind of invented it. And he predicted years ago that they may have a clinical use. A couple of years ago he said, we shouldn't call them mesenchymal stem cells because that implies that they only do a certain type of germ cell differentiation, which they can do everything. And this is the most important paragraph. This is the take home right here. This is what you need to remember. Kaplan said, the patient's own site-specific and tissue-specific resident stem cells will construct a new tissue when stimulated by the bioactive factors secreted by MSCs. So, before 2 years ago, people were giving MSCs from someone else, okay, from yourself. Very rare from your fat, from your bone marrow, and they don't last. I took a stem cell injection last November. See, I didn't feel anything. And, my immune system probably killed it within a day or two. So, some people have had some success with stem cells, but there is immune rejection. Whereas with this, the exosomes, there's no possible immune rejection. This is the secret sauce of how this is from the founder's own slide. So what you need to see is, blue is a mesenchymal stem cell. Red is an exosome, okay? And these are much smaller than shown, it's not to scale. So they get this one brand of stem cell. It's very hard to do, actually, to get it to grow and multiply to millions. And then they extract the exosomes, they spin it for a day at 100,000 G, and then they put it through a sterile filter so nothing bigger can get through. And then they suspend it in saline. They get out all the impurities, the growth media. They have fetal calf serum, which you don't want. And then basically what you're getting in this 5 billion exosome vial, I think is the product of like 1 million stem cells working for 2 days straight. So this is the recipe. I mean, everyone can do this, but it's not done well by everyone. When I get a shipment, this is what they send me. They send me a printout, and it shows size distribution. So we know this is the size of exosomes, about 100 to 130 nanometers. If we look at old high school geometry, the volume is 4/3 pi r cubed. So if a competitor's exosome is 70, then there's 137. I did the math. I just make sure they were right about that. It's about 7 or 8 times more volume. So bigger is better. Here's the CD9 marker, 63, also 81. They can test. It's present in large numbers. So this is the good stuff. If you take exosomes from someone who says they're selling exosomes, you don't really know unless they test them for these surface markers. Okay, there's our little friend, blue arrow. And these things, I have some here where I'm going to inject mom's knee. She tweaked her knee last week, so she's a little scared. But, I think she's pretty tough. I did it on myself without anesthesia. So anyway, so these exosomes are if you leave them out for a couple hours. Even I drove up to treat you one time. It was on ice. By the time it started to melt, it wasn't any good, so I had to detour and treat her. I carry them around at minus 109 dry ice because they have to be frozen. If they sit at room temperature, they're no good. So. And this is Dr. Ross's favorite little picture. He got a microscope about 6 weeks ago, which these exosomes are, again, 100 nanometers, very small. And he's able to radio label them. See the pink and blue? That's for CD9 and 81 or 63. So he can actually visualize what the exosomes are and if they're there. So he looks under his microscope. It's a $500,000 microscope. He can see that this is the real deal. These are the printouts they send us. These are the profiles. What you're seeing is proteins. And what I want you to see is room temperature. It all degrades. It's no good after a few hours. From amniotic fluid, you have a lot more particles, but it's a different kind of particle from what the baby's bathing in. And then on the right column is the MSC exosomes. This is some of the messenger RNA. Remember the clutch that can be read thousands of times, produce protein for weeks and weeks. It's a non-random distribution. That's the only point. And what you're seeing there is insulin-like growth factor. There's also other things which we'll talk about just a little bit about. This is a messenger, an RNA, all comers. So this will be messenger RNA and also blocking microRNA that we talked about. So you can see transcripts per million. I mean, there's a whole host of different notes these things are playing. And so this is for the scientists. I mean, this is interleukin-like. You can see 1 through 21. I mean, this is some pretty geeky stuff. What does it mean? We don't know. It's as though you were reading a score from Igor Stravinsky symphony. And you say, well, there's that note. What does it mean? It doesn't mean anything. It has to be played in the context of, chords, rhythm, meter, music theory, whatever. So these are some of the nice little, more sexy proteins that we know about. GDF11 is said to be an anti-aging agent. Granulocyte colony stimulating factor that just makes granulocytes grow. It's what they give if you're having a problem with chemotherapy. Stem cell factor, TGF-beta3, very important. It turns T cells away from inflammation, which is universally pretty bad, and it moves it towards healing. Vascular endothelial growth factor, another page, tissue inhibitor of matrix metalloproteinase. It's very important. It blocks people's collagen from being destroyed. Fiberglass growth factor. So, God, there's so many notes. It's like beyond what you talk about the music analogy I like to use a lot. We have, 88 keys, in Hindu music they have it like quarters. But it's a whole range of notes they can play, and the songs that they sing are only for their ears. Like, we don't understand how these cells communicate, and nor will we ever, really, to be honest. I'd like to shift now just to aging. These are not my slides. These are from Doug Spiel, who's like a real pioneer. Like, he's like a genius. He reads a ton of articles. I agree with some of what he says. I don't agree with all of what he says. He thinks aging is from genomic instability, which I agree with. The local stem cells mutate from number 2, telomere attrition. And that's that book that I wrote, the first one. My theory is that aging everywhere in disease is caused by stem cell mutation. When cells copy too many times, their telomeres shorten, and this causes genetic mutation. This diagram from my first book, Telomere Time Bomb, shows this very clearly. There's one engine driving all stem cell mutation, damage, and attrition. Another aspect of aging that we're just beginning to understand is epigenetic alterations. In my second lecture of the 9-part series on exosomes, I discuss this in greater detail. Basically, as cells age, not only is their DNA copied, but the software or the DNA methylation or silencing is copied. As we go on, more genes are silenced and less function is retained. Another aspect, proteostasis, is just a normal part of the fact that proteins degrade with time and they're not being replaced. In this next slide, we see some other broad topics of aging, including deregulated nutrient sensing. This speaks more to the aspects of how metabolism works and degrades. Mitochondria also get dysfunctional, although there is a bit of a red herring there because in the end stages of cell suicide, they actually are tasked with perforating themselves as a mechanism of melting from within. So when we see mitochondrial dysfunction and inflammation, it can often represent a cell trying to kill itself. Another aspect of aging is cellular senescence, and this is typified by what they're now calling the senescence-associated secretory phenotype, or the way that cells behave and message other cells when they're old. It's a broad general term meaning that old cells act funny, and in doing so they probably release weird signals, bad exosomes, and that leads to pathology. I think the only true things that we can say are that aging appears to be all of the above and comes down to two basic problems. There's stem cell exhaustion and there's stem cell dysfunction, represented by altered intercellular communication. In other words, what the cells are saying to each other is very complex, and it's degraded by epigenetic gene silencing, it's degraded by gene mutation and depletion. So mostly people, all of us included, like comforting lies, simple explanations, and the unpleasant truth is that cell-to-cell communication the ecology, the degradation. It can be described in general terms, but the specifics elude us, to say nothing of how to combat it. If we go to the other end and look at experiments done in terms of parabiosis, the interesting finding is that if you sew an old mouse to a young mouse, the old mouse becomes younger and the young mouse becomes older. Something about the circulating factors, which we now can guess are exosomes, adversely impacts one and benefits the other. So aging is not only in the cells, it's also in the messages that the cells are conveying through this senescence-associated secretory phenotype. And what we're really understanding is that the exosomes are how they're communicating and reprogramming each other. Now let's talk about the advantages of using exosomes over stem cells, which have been used in the last 10 to 12 years maybe pretty extensively clinically. The advantage of exosomes is that they go everywhere. They don't clump like stem cells will clump in the lungs, liver, and spleen. They can travel locally, yet they're carried systemically, and because they're so small, they cross the blood-brain barrier. They deliver the microRNA, which are crucial to expressing interesting phenotypes. We'll talk about a study below where they showed that the hypothalamus secretes some microRNA that are associated with the biological clock. Also, the mRNA, which is the clutch, allowing you to shift into a higher gear, so to speak. mRNAs can produce thousands of proteins because they are a blueprint that can last for weeks. They hone to sites of inflammation and repair. They're not perceived as foreign because in these versions of MSCs, unlike those from immune cells, there are no major histocompatibility complex antigens on the surface. They can never become new cells, and they can never introduce new genes. To introduce new genes is a process called, it's a process called transfection, and it can be done by means of retrovirus, reverse transcriptase. In other words, that's how HIV integrates itself into our genome. But there's no mechanism for these simple RNA messages that we know of to introduce the genes into the genome. Hence, you can't get cancer genes. You can only express maybe proteins for a couple of weeks. Whether that can reprogram cells is another issue entirely. They're easy to administer, store, and freeze, and there's easily controlled dosing, which is predictable, and also quality control is quite easy as well because they come from one source, a single clone variant. It's important to be aware that there are a number of contenders of exosomes in the market, but are they purified or are they just filtered for size and density? Most of the people selling these don't even know what the cells of origin would be, and they don't know what xeno-free culture means. That means that we don't have any bovine serum or other serum. These exosomes from a chimera are suspended in normal saline. Also, if you're talking about exosomes from bone marrow, that's usually cadaveric. Do you really want the exosomes from somebody who has already died? It would depend on how old they are and whether they had accrued any illnesses or mutations or weird expressions of their genes along the way. Also, some of the exosomes on the market are smaller. These are robust, 120 to 130 nanometers in diameter. Again, maybe there's more product inside if they're larger. I'd like to talk about a few interesting studies shown in rats, and they use human exosomes because that's what's available and because rats and humans express pretty much the same genes. In this study, they showed that if you give a rat a stroke by tying off its middle cerebral artery and then give it tail vein injections, the rats significantly improved in their recovery as opposed to those who received no treatment.
Same thing with heart attack. Rats were given heart attacks by tying off their artery to their heart, and tail vein injections of MSC exosomes from humans were given, and all those rats that were treated had improved cardiac function and reduced sequelae of the damage. Finally, they gave it to animals. Actually, these are patients with stage 3 and 4 chronic renal disease. They were given MSC exosomes, and their kidney function improved. The study I was alluding to about hypothalamic stem cells showed that one of the microRNA, or blocking RNA, if you will, is originating in this area of the hypothalamus, called the medial-basal hypothalamic region. It's important to realize that we do make new brain cells, unlike what they taught us in med school. The hippocampus makes 10,000 new brain cells every night when you dream in deep sleep, and this is how we form new memories. In the ventricular zone of the lateral ventricles, these are producing cerebrospinal fluid. They're also progenitor stem cells that copy, and it's important to note that hypothalamus is the aging clock. It controls the pituitary, which controls all the hormone and endocrine systems. But the hypothalamus is also the center of our level of arousal, drive, aggression, interest, sleep, biological clock, and that inflammation to it will damage the hypothalamic cells and possibly speed up aging. It's possible that aging involves inflammation over a lifetime, and that's how being inflamed and stressed both impacts immunity and ages us faster. By implanting neural stem cells which were resistant to inflammation in mice, they showed that there was an anti-aging effect. And also in this study showed that these blocking mRNAs produced by the healthy neural stem cells produced a ton of these blocking RNAs in the hippocampus, which may be why we can stay younger, and hopefully some of the growth factors in the exosomes will promote this as well. Clinically, we turn to some of the fun cases. Here are some slides showing vitiligo, which is an autoimmune depigmentation condition, and I think you can see some repigmentation coming in after treatment with IV exosomes, not into the skin. You can clearly see on the right there are new patches of pigment coming back. Here's a case of rosacea, which you often see as redness and or raised bumps and rashes around the cheeks and nose, and remarkable results only 2 days after intradermal injection of MSC exosomes. The uses for exosomes in musculoskeletal therapy are broad, and they're based on these 4 things. They are creating new muscle or musculogenesis, bone osteogenesis, nerves neurogenesis, and vessels to supply them, angiogenesis. This slide shows simply that as we age, arthritis becomes more common. It's a little more common in women, but in men and women it increases over time. And I think it's good to think of it just in terms of brake pads. Brake pads undergo friction, wear, damage, incomplete repair, the damage to the matrix proteins, and it's shown in this one study by Price et al. That the telomeres in the cartilage near the lesion of the same joint are shorter, again supporting my idea that telomere attrition is depleting local progenitor stem cells, both in terms of quality and quantity, and that is why we manifest arthritis. Here are some of Dr. Spiel's pictures from a 70-something-year-old woman. The joint space between the bones, which is black, is narrow, and that's where the cartilage is. And he suggests that it gets wider after treatment. This treatment was simply exosomes into the joint space. Here's an interesting case of a patient with inflammatory bowel disease. His ulcerative colitis caused pain 5 out of 10. After treatment, it went to 0 for 3 days and remained 1 or 2 for weeks after that. Remarkable result. Arguably, the intraperitoneal injection was a more direct and possibly more effective route than intravenous. Here's a case of a 19-year-old man who underwent a boating accident in August of 2015. He had tried stem cell infusion, and he underwent a cervical fusion of C4 to 6 to stabilize the region. You can see the white area of damage. After exosome injection, he had some improvement. These videos show his ability to control his torso while on a bike before.
The only thing is kind of starting in the middle thing now.
And then after treatment.
Wow. Wow. Oh my God.
And you have control of it, right? You can turn.
Yeah, exactly. All the way, Sean.
Another fun case is a 28-year-old man who wasn't sure he wanted to do exosomes, didn't know if they were safe. He was Asia Class C, meaning that he couldn't move 50% of his muscles against gravity. He underwent high cervical spinal injection and IV exosomes. Interestingly, he refused to do it until Dr. Spiel could prove it was safe. As he said, how am I going to do that? He said, well, if you think it's so great, why don't you prove you can grow back my hair? And so this shows he did grow back a bit of hair, and then he allowed himself to be injected in the spine afterwards. And this video shows him bearing weight and even supporting his weight, which moves him from a class C to a class B. He's able to stand up against gravity.
So we're going to balance on our own for the second time. Yeah, I've got it. All right. So this is me just using my arms to balance with Tony.
In conclusion, I just want to say that as we age, our stem cells become altered and genetically mutated, epigenetically less capable of expressing the proteins we need. The science and the experiments that are coming out show that even older cells can still sometimes respond to the younger exosome signals, and that's a very exciting finding with broad repercussions for how we treat aging and illness in the future. The use of exosomes is under a patient-physician relationship. You need to be fully informed about the risks, benefits, and unknowns. You might want to wait for the FDA, but even if it is FDA approved, it'll be for one indication at a time. It might be approved for advanced Parkinson's, and if you have a bad knee, that study may or may not ever get done, so you would have to, again, take it in an off-label way. I would stress that quality control and consistency is very, very important, as is maintaining a frozen chain of command. I would suggest that quality control and consistency is very, very important. The science is very precise. It is an art, and if you don't keep them at least minus 30, they degrade. It's important to have good clinical experience and judgment and to learn from your mistakes and to always be learning, and that is something I indeed strive to do. So thank you for coming. I hope you got your parking validation, your samples of recharge, and please leave your email so I can include you on my lists. After the break, we'll have a lecture called The Clinical Use of Exosomes, and we'll show an injection on my mom's knees. So mahalo, thank you for attending my lecture, to Akimi Malin for helping me organize and introduce me to all these wonderful people. such as Wally Inuma, who introduced me to his rolfer, the famous Marianne Wassel, who's booked out a year, I understand, and to Mary Lou Palmer, my wonderful friend who runs the supplement and health store on Nimitz Highway called Designs for Living, where you can also purchase my recharge locally. Finally, thank you to Mrs. Ariyoshi and to her friends for attending the lecture as well. I'm very happy to show here my newly printed wall certificate for the state of Hawaii, as I am licensed to practice medicine here, and I look forward to meeting many of you and helping you and your friends and your loved ones. Thank you so much. Bye-bye.
I want to tell you, oh, just how much I love you. Yes, I do. So come with me to the sea, my love. Do you remember when we met? Oh, it was the day I knew you were my pet baby.